Thank you. Yeah, sorry I couldn't be there. People I was intending to be, but didn't work out that way. And I put this ambitious title together, but I'm just going to summarise a bit of work that we've been doing in some published PhD work. I'll mention some new stuff that we've started on, but not in too much detail. So I thought I'd make a couple of comments about the collaboration with LEPSE. I had a look through my computer and found these slides from 2004. I think the first time I was encountering the team was probably in around 2000 or 2002 at a Chapman meeting, I think in Paris. But we were at this Generation Challenge program meeting in Montpellier, organised by Francois and team. And we were talking about this work that we've been doing. And I guess one of the quotes from the first time I really had longer interactions with Francois was, we were talking about this work on drought environment types, environment characterization, and so on. And I had to tell him what AJR stood for, which was the Australian Journal of Agricultural Research. And then I discovered that INRA didn't subscribe to that. CIRAD didn't either, for that matter. So we realised that it was going to take a while to propagate the research without publishing it in places that people knew more about it. So I guess that was one of the functions of this small story in plant physiology, which I think Yvonne referred to indirectly, talking about plant size. So at that same meeting, Francois was talking about the same kinds of things that you're hearing to talk about today, but in a more introductory way and still with the nice yellow colour. But describing the messy phenotype from the messy methodology, but being able to build these nice models. And that really is what started a lot of the relationship through GCP, whereby we published some of these works in some journals that apparently people started to read. And this is actually a screenshot of the poster that Corinne put together 3 years later when we were starting to build the models of connecting genotype to all of the LER models that have been developed at LEPSE, and particularly from the work from about 1996. So that's where it started, and then I thought I found some interesting photos, and it's nice that I was immediately after Laurence, and unfortunately I can't hear whether he's laughing in the room, but I'm hoping everyone can read this because my French isn't good enough to pronounce it to you. But we did start to worry about the gastronomy in France, although Francois was not too worried about it on that night, and but Al Doherty apparently was. But this was actually a different restaurant, so it wasn't the one we were. So where do we hope to be going in 2024, which would be sort of 20 years after that? And I just put these words on here as, reflecting what Ionis was saying, that we're really trying to combine and bring in machine learning and additional types of statistical models and integrate them with their physiological models to improve precision. And part of what we're trying to do today is trying to improve how well our models can describe what we see in the trials, so that we can interpret that in statistical models, whether they be classical models or mixed models, or they be some advanced versions of those. But the utility that we often see in machine learning is that we can improve precision of what we're measuring, even if we don't have that same degree of power and prediction that we might have with a physiological model. I'll talk a bit more about that. So the main motivation for a lot of this work is a project called Inviter, which I shamelessly stole the name for that, Innovation in Variety Testing. There's the Invite project, which is Invite with an E, and we decided we were lazy about describing the project and call it with an A. But it's important to know that the initiation of the EU Invite project, which is led by INRA but involves, I think 27 or 28 organisations, and particularly the engagement with bargaining was really important to motivating Australia and GRDC to invest in this project. So we have these three types of works that are going on in there: augmentation of data analytics and simulation. I'll talk more about some bits of the simulation work than I will talk about some of the others. But in the Inviter project, we're looking at over 600 trials per year in multiple species for the variety testing across Australia, which is run by GRDC. And we built a series of trials and measurements on top of those over the last 3 years to do extra measurements to try to get to a better regional prediction of performance. So here's an example of one of those trials. It's from 2020, and you can see a screenshot here. It's captured in Hyphen's analysis system. We were using drones to capture data from this. And here's a national variety trial. So there's actually 3 reps there, 1, 2, 3 of about 40 varieties. And we augmented that with SatCalc trials, which were the same variety, trying to look at what the satellite was seeing, and BioCalc trials where we had multiple densities and varieties and we were taking extra measurements in these. You have to keep in mind that while we were doing this at 30 to 50 sites out of the about 120 sites that were there for wheat, these sites are often remote. I mean, they're often 200 kilometres from the people who look after it, and they might only be visited 4 or 5 times in a season. So you can't intensively monitor these. The only thing we can use to intensively monitor really is satellites and cameras that we might install in the system and sensors that we install in the system as well. So we're trying to assess a few different things for Inviter. One is how to improve trial quality, but another is how to monitor these things in some efficient way and make use of that data. So here are one of the benefits of living in a somewhat droughted country is that we do get a lot of high quality satellite imagery. So you can see that through the year we get quite a lot of nice pictures from Sentinel-2, thank you Europe, and we're able to use this data very well for yield forecasting. I won't talk about that tonight, or today. The kind of work that Andries Potgieter and Graham Hammer have been doing over the years in forecasting wheat yields for Australia depends on these tools. But we're looking at it at the trial level and trying to understand environment characterization. So really the rest of this talk will just make some mention of how we're trying to improve the way that we measure environment characterization using sensors and models. So to do this, we're using the integration of crop growth models with radiative transfer models. So the ones that most people would know about will be, well, the ones that we've been using, AppSim associated with ProSale or versions of ProSale. And the way that we've been doing that is having AppSim or the crop growth model set up to run G by E by M experiments, creating outputs from that, and then feeding that information and additional information into a radiative transfer model. So we calculate that to estimate crop canopy reflectance. So effectively you're using this pipeline to create reflectance models, and the interesting thing for us is then can we build mathematical models that can invert this? So how do we get back to LAI, or how do we get back to anything that can be calculated in the crop growth model? So that's the first point. And the second point is, can we do this in a completely synthetic data set and then transfer the mathematical inversion to real world data sets? So we're certainly not the first people to try and do this. It's been done with radiative transfer models for a while, but what we were really trying to do was use a crop growth model to impose biological constraints on the sets of parameters that you train the model with, and to demonstrate that that was valuable to do. So if you want more detail on that, have a look at the paper from Sophie Chen, or Kim and Chen, in Plant Phenomics, because what we were able to show, or what Sophie was able to show, is that when we use a crop growth model to drive the RTM you don't sample all of the parameter combinations that could happen in an RTM. So the published papers up until then would sample all of the variables that could go into a radiative transfer model, so all of these inputs, and then create a big data set of canopy reflectance, and then try to invert that with a machine learning model. But some of those RTM inputs were ones that were physiologically impossible. So you might have high combinations of LAI and SLN that were never going to occur. Whereas if you ran it through a crop growth model first, it would eliminate those. We were able to show that when we did that, we improved the quality of the model. So Sophie and Peng-Chen Hu, who was a postdoc working on this topic in the Inviter project, put together the AppStim bare soil model and ProSale framework. So Sophie and Peng-Chen were looking at how to improve the model a bit more. So when we're looking at canopy cover not being complete, we have to account for the bare soil. So we had to look at how we would capture that effect. And so these were really just refinements of the connection between crop growth model and the RTM, which is the ProSale model. The important thing here is that, AppStim is not a 3D plant model, so it's not simulating that. So we're estimating cover with conventional methodology based on extinction coefficient and things like that. So we use it in two ways. One is to calibrate cultivar environment parameters from the trial network. So we've got daily weather data, we've got to infer what the soil attributes are. We've got the management practice because we know what it was. We've got to infer what the cultivar parameters are. And so these two sets of parameters, we create a big matrix to search through and look at using numerical optimization to do that. The flowering dates, we've already optimized all of those for the cultivars based on thousands of observations in the national variety trials already. So we've already optimized the way that we estimate those. But for individual trials, we then have to work out what the soil attributes and cultivars attributes look like. When we run that through the combined model, we can estimate crop traits, but we also can estimate canopy reflectance bands. And then the idea is that we can take NDVI data, whether it's been collected at the UAV level with a drone or whether it's been collected from a satellite, and we can run this backwards and try to estimate characteristics of the varieties and of the crop traits themselves. So this is the way we've been running it. Excuse me. To take observations from about 6 years worth of trials, 13,000 observations, and estimating flowering data across the country. So this allows, it doesn't look fantastic, but these lighter coloured blue squares that you see here have very few observations. They're sort of less than 20 observations. So really focus on the yellow and red numbers, which have a huge number of observations across all of the varieties. I can show you the data for each. We can fit them very well across environments. So we can predict flowering time quite well. But what we can't do is we can't measure all of the soil profiles that we need to measure. And so to do that, we have to run some kind of simulation process. And then the assumption is that there's a uniform soil across the trial, and we should have all of the varieties working well with the same soil. So that's one way of addressing the issue of multiple solutions. So that allows us to simultaneously fit these G parameters and E input variables. So we've tested this against those biocal trials where we actually took some biomass samples. This is at the trial level, so these are the single dots for the whole trial. And these dots are for individual plots where we had a range of densities and varieties and across a large number of sites. So we could predict it that well. Then we would connect it to ProSale and try to simulate what the satellite would see for the whole trial. And we could get a reasonable estimate of the NDVI from Sentinel from using ProSail. So basically, we're trying to work out how to align the pattern, the spectral pattern between the model and the satellite. And so that means that we can either do a crop trait based approach, and I've already explained why in Australia that's very impractical when the trials are 200 kilometres from where you want to be and you just can't go and do all that biomass sampling. How well can we do this using spectral index data and our prior knowledge of the varieties, and we can do a fairly good performance. This work's not quite yet published, so that takes us to basically more reliable simulation to identify the environment types, because these environment type patterns, so this is the stress through the seasons centered on flowering, so he's flowering here. If you classify all of the trials across the NBT for the last 6 years, you'll get this kind of pattern. Sorry, this is from Corinne's work in 2011, but we were looking to see how often these patterns occur, but we can better inform each trial with weather and satellite data. And then we're trying to take that a bit beyond the environment types by considering water and nitrogen stress, cold stress, heat stress. And so this is the work of Javier Fernandez, who's been reclassifying all of the ag zones in Australia. And we're looking at the year-to-year variability in those. So for how frequently across the country do we see low water stress, moderate water stress, or high water stress, and how much does that vary? And you can see we have a really wet year like 2016 where we have a lot more low water stress, but in general, it's a pretty big proportion of reality. So these are for the national variety trials themselves. So the National Variety Trial Network, all that work that we had done with Corinne a few years ago, that was with some subsample of sites that didn't quite represent the whole network that this does. So the second part of it is really using the methodologies that I explained just before to monitor the crop growth dynamics. So feeding all this information through AppSim, training it up, and then trying to use a machine learning model that accepts weather data and observed satellite data, and we try to back calculate the LAI and the biomass. We've already estimated the phenology quite well because we know what variety it is, and we've already got a good model of that. So that means that we can use a machine learning model to start to look at the dynamics, and it has great utility combining this biophysical model with an RTM and being able to use machine learning to take observed data and predict crop traits. And so if you want to see some of the work on that, sorry, I'm going to skip ahead a little bit. Sophie's published this in that paper. So this is just a commentary on what she did in that work, where we either took what we called R data, which was random data. So for example, if you looked at LAI and CAB, someone building a machine learning model using ProSail would, by default, would sample this entire space. But if you look at this space here, this red space, this is what we call a sort of biophysically constrained space. So AppSim would say, well, you know, this space cannot exist. So if you remove that space from the training of your machine learning model, it improves the performance of the model, and we're able to show that that's the case. And this is some of the work on predicting leaf area index at different stages. And I just ask you to go and look at Sophie's paper for more detail. So in conclusion, I think these hybrid models are improving our understanding of the in-season environment. So perhaps in our ugly slide that I had in 2004, we should add and ML into the set of models. And certainly they don't replace our physiological knowledge, but they've got a lot of utility improving this precision and allowing us to develop high quality machine learning models. So we think these synthetic data sets have a huge value, and you've already seen several talks where people have shown in 3D imaging and that kind of work that machine learning models have a lot of utility, but we think that they have a lot of utility in a process-based approach as well. I didn't see the talks yesterday, I'm sorry. I'm assuming that Graham and Fred might have mentioned some of these other types of models, but how do we still retain that level of prediction, because the machine learning models don't necessarily allow it? Although we can use them in a mode where we might use forecast weather data, but we might also look at, well, let's forecast the satellite index and then run the machine learning model. And we're testing some of that at the moment to see whether that approach works as well. And I better stop there because I think I've probably used up my time. I do want to highlight Sophie, Punchen and Javier, who are the main contributors to this work. And just to mention that, you know, a lot of this is going to be part of a new analytics initiative in Australia called the Analytics for Australian Grain Industry. And we're starting to set that up, and I'm hoping to visit people in Europe in the next year or so and talk a bit more about what we want to achieve there. Thank you.